Okay, so this is chapter 12. Oops, sorry guys. Whoops. This is chapter, hold on. Chapter 12 in the 9th edition and chapter 13 in the 10th edition. So there are lots of things we have to take into consideration when we try to figure out how to control microorganisms. So some of the things that we have to take into consideration is how fast organisms can actually die. So, wait a minute, my pen's not working. Okay, how fast they die. Okay, how many organisms are present and how they're arranged. So it depends on the amount of biomass. So if you think about a biofilm, so when things are in a biofilm, right, they're in layers. So when they're in those layers, it's harder to get, and you have to have an antibiotic or antiseptic or disinfectant that's going to be able to penetrate those layers. Okay, another thing that's going to influence it is organic materials. So is the pathogen in vomit? Is it in blood? Is it in feces? Okay, and then we have to look at, and this one to me seems kind of common sense, right? We have to see how susceptible it is to whatever you're trying to kill it with. Okay, and all those things are related to time. Okay, so how long does it take, what the pH of the solution is, what the temperature is, and the concentration of the antimicrobial agent. So all this is I feel like it's kind of common sense. Okay, now here's some vocabulary. So static. So sometimes antimicrobial agents are referred to as static, and sometimes they use cidal. So if something is static, what is happening is, if you look at this growth curve, right? So the black line is the normal growth curve, and then if you look at the yellow or the blue, right? The blue is flat. So we're not killing off all of the organisms, but we're inhibiting their growth. We're slowing their growth. Whereas something is cidal, right? Cidal, we're killing it. So here's some examples. So bacteriostatic, fungostatic, virustatic. Things can be bactericidal, virucidal, and I put in here suicidal and homicidal, so you would relate those two things, relate cidal to death, that things are actually dying. Okay, now more vocabulary words. So whenever we start to talk about sterilization, so sterilization is removing all of the microorganisms. So it's removing everything on an object or in like the media that we make. So we want to get rid of everything. If we disinfect, what we're doing is we're reducing the number of pathogenic organisms so that there's not enough of them to cause disease. So it's not sterile, right, but it's disinfected, so the number of pathogens are reduced. Whoops, hold on. Okay, so now categories of agents are, we have a little laundry list. Okay, so the first thing is antiseptics. So antiseptics, it's important to realize that they can be added to living tissue. So you could put them on living tissue and not damage it. I'm trying to find a better stylus. You can put them on living tissue and it's not going to damage the cells. So they're non-toxic to our cells, the human cells. Okay, but they're going to kill or inhibit the pathogens. So they could be static or cidal. Okay, most of them aren't strong enough to destroy spores. So things like Clostridium could still be there. Okay, so here's some examples. So one is hydrogen peroxide. So the symbol for hydrogen peroxide is H2O2. So this is hydrogen peroxide, and I have bottles of it here. Okay, what you buy at the CVS or Walmart is 3% hydrogen peroxide, but you can get it a lot stronger. If you have a stronger strength, it can damage tissue and cause chemical burns. Okay, ethanol is another example of an antiseptic. 70% is actually the most effective ethanol. well, between like 40 and 70. Okay, so 99%, it evaporates really fast. So it's not enough time to disinfect, where the 70% has some water in it. So that's going to, it's going to stay a little bit longer. It'll break down the plasma membranes and help break down proteins, denature proteins. Okay, now disinfectants we use on inanimate objects. Okay, so they're usually at a higher concentration and they could damage tissue. So you don't want to put something like phenol or bleach on tissue. It'll damage it. Okay, another thing about disinfectants is they can remove toxins from the surface. If the microorganisms are dead, they can actually get rid of the toxins too. Okay, they don't usually kill spores, but bleach can break down Clostridium spores. Okay, so examples: 5% bleach. So the active ingredient in bleach is sodium hypochlorite. Okay, so this chlorine part is going to be the active ingredient. Okay, most bleach that we buy, it's 5% sodium hypochlorite. Okay, then we sometimes dilute it after that. Okay, then phenol is one of the first disinfectants. So Lister is the one that came up with that. So remember, Lister is considered the father of antiseptic or sterile technique in surgery. So he actually had kind of like a diffuser, and he put carbolic acid in it and spread it all throughout the room, kind of like create a mist of this phenol, which is actually very dangerous, not good. It's a carcinogen. It can cause chemical burns if you get a bunch on your skin. But it disinfects. It kills everything. Okay, other disinfectants. So ethyl alcohol and chloramine. So I just have some little pictures. You guys don't have to memorize the chemical structures, but these are just different things that break down, destroy bacteria. Okay, so more about alcohol. So there's two different kinds of alcohol. So there's isopropyl alcohol, which is rubbing alcohol, and then there's ethanol. So both of those work. The optimum concentration is between 60 and 90%, and they say that 70 is the best. So if you look at your hand sanitizers, they're usually about 68%. If it's less than 50%, it doesn't work. It's not good. But it can kill the organism that causes tuberculosis. It can kill fungi. It can kill viruses or denature viruses, inactivate viruses, but doesn't usually kill spores. What it does is it denatures the proteins, it dissolves plasma membranes. 100% it'll dry out the cells in the 99, but it's not on there long enough to destroy the proteins. Okay, hexachlorophane is an example. They call it PhysioHex. It's an example of a disinfectant that is kind of bad. Okay, so it's a really, really good skin disinfectant. If you put it on your skin, it will kill staph and gram-negatives. It has phenols in it, so the phenols in it break apart the cell wall and disrupt the proteins. So it's going to kill organisms. They used to use it when babies were born in the 60s. They would scrub them with PhysioHex to clean the skin. So they were like disinfecting the babies, and it actually got absorbed through the skin, and it affects the central nervous system. So it caused problems with the babies. They had learning difficulties. Okay, it is available. I mean, they still use it for skin disinfection, but it's not in infants anymore. Okay, now when we evaluate disinfectants, what we do is we compare it to phenol. So Lister introduced phenol as a disinfectant in 1867. So since it was like the first one, he considered it the gold standard. So we compare everything to phenol, even though phenol might not be the best anymore. Okay, so if something has a phenol coefficient of 1, that means it's as effective as phenol. So if you look at this chart, so phenol compared to phenol is 1. Okay, so phenol is effective against staph and salmonella. Okay, now some other things, right? So chloramine, cresol, ethyl alcohol, you can look at those and Lysol, compare them with phenol. And pretty much everything is stronger than phenol except for peroxide. Okay, so we use the phenol as the gold standard. So they call it the phenol coefficient. Okay, so we're doing this in lab. So one way to evaluate how effective chemicals work is this thing called the filter paper method. So basically what we do is we're going to take a big petri dish and we're going to make a lawn of bacteria on it. So all of this is bacteria. And then we'll add some filter paper discs that are dipped into to different chemicals. And then we're going to look for a zone of inhibition around the ring. So the bigger the zone, the more effective the chemical is. Okay, another test, and I don't know that I have this written out well, it's called a use dilution test. So what they do is you have the test bacteria, okay, and it's growing in a tube, and you dip these stainless steel little cylinders into the broth and get bacteria on them. And then we take those, and so this is the chemical. Okay, and we take them out, we rinse them. Okay, and then we're going to put them in the chemical. Actually, we don't rinse them before we put it in there. We take them out of the broth, we put it in the chemical, then we rinse. Okay, and then we'll put them in a tube with broth, and we'll look to see if the organism actually grows. Okay, so the goal is to see how dilute you could actually get the chemical and to have it still be effective, but make it have the best cost benefit ratio. So if you can use something more dilute and it's less hazardous to people, that's better than having something super duper concentrated. I hope that makes sense. Okay, so you take the stainless steel discs, you put it in the broth, then you'll put it in a chemical, and what you're trying to do is trying to see if the chemical will kill the organisms. Okay, then you rinse it off, okay, and get the chemical off, right? And then you put the stainless steel disc in another tube of broth, and you shake it, and you try to see if the organism will grow that you initially started, or if it's dead. Okay, so here's a list of things for an ideal disinfectant. Okay, so it has to be fast acting, even if there's organic solids there. So if there's vomit or blood or tissue. Okay, it should be effective against a wide variety of pathogens, but it shouldn't damage the tissue or be poisonous to the person. Okay, it should penetrate the material, but it shouldn't damage it. It should be stable, so long lasting, have a long shelf life, and easy to prepare. Okay, it should be inexpensive, easy to find, and then no unpleasant odor. Okay, so I have a couple little practice questions. So one of them says, are there degrees of sterility? So no. Sterility is like either you're sterile and you don't have any organisms on you, or you're not sterile. So there's no gray in sterility. It's a yes or a no. And then this question is asking about phenol coefficient. So product A has a phenol coefficient of 2 and product B has one of 0.5. Which one's better? So A is better because it has a higher number. Okay, now I'm going to stop here and